Okay, guys. In the last two sections, we have discussed DNA extraction, how we get DNA extracted out of nucleus, out of cell membrane. And the last section, we were discussing electrophoresis, DNA electrophoresis, and we have completed electrophoresis with subsequent process called blotting. And we have discussed that there are three types of blotting: southern, northern, and western blotting, and so on. Okay. Today we will talk about PCR and the importance of PCR in clinical diagnosis. PCR, short for polymerase chain reaction, okay? So there is an action of polymerase, and this action of polymerase repeats itself to reach a copies or a variety or a massive or a large amount of DNA copies, okay? The story starts with, let's imagine that you are looking at the sky at night. All what you can see are stars, right? And the moon, okay? Such stars represent themselves as points, okay? So you are looking at the star and receiving and perceiving them as points, but actually such stars, everyone is maybe as large as the sun, right? How you can figure out such facts? By using telescope. By using telescope to magnify the star you are looking at till you reach its actual size, or can determine and estimate its actual size. And the same, likewise, with cell organelles, nucleus, mitochondria, and Golgi, okay? How can we identify such objects, such organelles, by using a microscope to magnify the subcellular structures, right? The same way here, I'm looking for a specific DNA segment with a very large size, with a tiny size. So I need to magnify the size of such particle or size of such segment. But as a matter of fact, in clinical practice, it's impossible to do this, to magnify chemical structure as nucleotide in size. So what we will resort to is to magnify the sequence, but in numbers, okay? To make copies, identical copies of the copy of interest I'm looking for, if they are existent or not in my sample, and if they are magnified into a significant number of copies so that I can figure out, I can see, I can detect their signals, then I conclude that there is a fragment or there is my DNA target within the sample. But on the other hand, if the magnification or if the amplification, if you could say that, amplification to increase in copy number, if such an amplification didn't or doesn't occur, then I conclude there is no DNA or there is no such DNA segment at the very first start, okay? So this is the principle of polymerase chain reaction. Meanwhile, before they have invented PCR and they determined the process, the determinant of the process, there was such a thing called molecular gene clone, which in brief, they take the gene of interest with sticky ends, and they make use of the plasmids inside bacteria, and they treat such bacteria with the same restriction endonucleases to cut off into the same sequence or the same DNA or nucleotide sequence order, likewise the gene of interest. So the result will be two segments, one of the plasmid with sticky ends, complementary to the sticky ends of the gene of interest. The next step will be to apply such segments into bacteria to be sticky or to bind to plasmid. And I allow bacteria to proliferate, and meanwhile bacterial proliferation, there is also proliferation for my segment. In the end, I can either use such DNA segments themselves or to let DNA segments to be translated into protein, and I make use of protein as drugs. Likewise, the common example, the common instance of insulin production. Insulin is produced such way. Okay? What is the difference between molecular gene cloning and PCR? Molecular gene cloning make use of the bacterial replicative machinery to make a lot of copies. But PCR occurs in vitro. So all I need to make or to do perform PCR is to supply the required material for such PCR reaction to occur. So I need to supply with primer, the probe, the starter that will determine and point out to the DNA segment of interest. So this is number one, the primer, okay? Number two, I need to supply the synthetic nucleotides. And I need to supply and provide the reaction with a kind or a special kind of polymerases, okay? So these are three things. The thing is like, you know, you are building a condominium or you are building, construct a building, okay? So what do you need to construct a building? You need bricks, right? You need workers, right? And you need someone who points out you should build here, okay? You should build here means at the DNA of interest, the DNA sequence of interest, the gene of interest, okay? This is the primer, the action of the primer. And instead of bricks to construct DNA segment, an identical copy of DNA segment, we need synthetic nucleotides, okay? And instead of worker, we need polymerases, okay? But there's a problem. The problem is I can't reach the specific segment while DNA is coiled as double-stranded. So I need first to, you know, do unwinding to the double-stranded DNA to expose every single strand with the genes contained within to the polymerase and to the primer at first to point out to the DNA of interest, okay? To do such a thing, I have two options. Number one, to apply or to supply my mix with helicase enzyme. Likewise, the bacteria helicase. Helicase do, you know, break down the helix. Helicase break down the helix, right? To cut down or cut off hydrogen bonds to separate double-stranded into single-stranded single strands. But if I apply helicase with polymerases, their actions will be contradictory. They will conflict or they confront each other. Polymerase itself will add nucleotides, and meanwhile helicase, he destructs the nucleotides polymerase added. It's a problem, right? So I need another option rather than helicase enzyme, which is, like we have discussed in the theoretical curriculum, we have temperature, okay? So I will apply temperature in such a way that first will do denaturation to the double-stranded DNA to expose the specific segment, okay? So that the primer will find out its target, will point out to the startup sequence, okay? And then I will do some cooling temperature, rich cooling temperature to 55, relatively cooling temperature, 55 degrees or 55 Celsius, okay? At 55 Celsius, the polymerase will take a standby position at nucleotides. The synthetic nucleotides floating around will be closer to the DNA to be built, and the complex are ready for polymerization. Once I rise the temperature to 72 degrees, the work starts off. And polymerase make use of the floating nucleotides to add nucleotides, each at one or every nucleotide, step by step, to reach or to complete the whole newly formed strand. Okay? So again, temperature is the key word of polymerase chain reaction. By increasing temperature into 95, I do unwinding of double-stranded DNA to export the single strand to primers. While dropping temperature to 55 Celsius, I make a standby position. Polymerase at the primer in its place, and floating nucleotides are getting closer. By rising temperature back to 72, this is an optimal temperature for polymerase to function. And it adds nucleotide at a time, each one at a time, to complete the newly formed strand, to duplicate the gene of interest. Okay? We have something left here to discuss. What are the characters of such enzymes, tag polymerase enzyme? Here, tag polymerase, okay? Tag polymerase enzyme, it is the thing that allow or make such a thing like PCR to occur. Because without the discovery of the bacteria, which is called Thermophilus aquaticus. Thermophilus aquaticus, they are species, they are living in aquatic water, okay, aquatic lakes, and aquatic lakes are exposed to high temperature. So such enzymes are tolerable with, or can tolerate the high degrees, high temperature, okay? So they can tolerate the change of temperature from 95 degrees, for example, 95 Celsius, for example, to cool down relatively up to 55 and to rise back the temperature to 72. They can tolerate such modification in temperature, okay? And their name is Thermophilus aquaticus, named after the bacteria they produce and they are discovered firstly, okay? So they supply Thermophilus aquaticus with the master mix that contain synthetic nucleotides and that contain primers to point out to the start of the chain of interest, okay? So this is the whole story of one cycle of PCR. But what is it about the other cycles? PCR is called polymerase chain reaction. There's a chain of reactions, okay? There's not a single reaction, there's a chain, okay? So there is no one cycle. What I have discussed by rising and then cooling and rising again, it's one cycle that results in 2 copies. If I start with 1 copy of G, it will result or it will lead to 2 copies. Likewise, you see here. I have 1 copy, 2 copies, okay? And if I run a second or a next cycle, it will result into 4 copies. 2 and 2, 4 copies. And if I run another cycle for 4 copies, I will reach 8 copies, okay? There's something here that is worthwhile to be noted, is that the amplification doesn't occur or doesn't happen, by stepwise approach. Not like 1, 2, 3, 4, 5, 6, 7, 8. It's not like that. It's 1, 2, 4, 8, 16, 32, 64, 128, and 256. Okay. So this amplification is called, they're calling it exponential amplification. Exponential amplification. Exponential amplification, all this is a repetition to what we have discussed, we can revise it at home. exponential amplitude to reach a large number of copies within small number of cycles, okay? And they have decided on a simple rule, that's it, 2 to the power of n, where n refers to the number of cycles. But there is one condition. I start the reaction or the first reaction with one copy, okay? But if I start the reaction with 2 copies, then I can multiply the 2 to the power of n by 2, okay? Okay, so 1 copy will 2 to the power of n. 2 copies, if I start the reaction 2 copies, if there is 2 copies at the very first start, then 2 multiplied by 2 to the power of n. Like if I run 2 cycles, okay? If I run 8 cycles and start the reaction, first reaction with 1 copy, so the calculation will be 2 to the power of 8 cycles, 2 to the power of 8, okay? And so on. So this is the way I calculate the number of copies that result from PCR as regards the number of cycles. What is it about RT-PCR? It's a simple thing if you understand PCR. If I have, in some scenarios, I need to amplify RNA rather than DNA. So what I should do is to first reverse the transcription of RNA to what's called cDNA, complementary DNA, and then run the same process of PCR, the conventional PCR, the conventional process, okay? So it's pretty easy. RNA, then cDNA, and cDNA will be treated as the conventional PCR, okay? This is a summary of RT-PCR. And RT is an abbreviation for not reverse transcriptase, real-time PCR. RT, real-time PCR, not reverse transcriptase, okay? In spite of, I use the reverse transcriptase in the process, right? But the abbreviation is for real-time PCR. So, from this slide, I can say what are the scenarios that obligate us to amplify RNA rather than DNA. Some viruses contain in their genetic makeup RNA, like HCV, hepatitis C virus, like HIV, AIDS virus, like coronavirus, 3 or 4 years ago, okay? We needed to amplify specific sequences on such RNAs to figure out if this virus is existent or not, if this virus is or has infected such patients or not, okay? So I needed to look for specific targets on such RNAs. By this concept, I needed to reverse the transcription of RNA into DNA first and run the same process of PCR, and the result will be the diagnosis of such diseases. So if you are asked about the usage or the purpose of PCR, your answer will be first, diagnosis of infectious disease. But there is one condition or 2 conditions in such diseases. Number 1, the types of culture or types of infectious diseases that are difficult to culture, like what? Like the virus, as I said, HCV, corona, HIV. Why is it hard to culture or to cultivate viruses? Because viruses are known as obligatory intracellular organisms. They are not bacteria. Bacteria, if I give bacteria some nutrients, agar and some sugar, they will thank me and flourish, okay? But viruses need, to grow, need cells to infect first and to make use of the genetic makeup of such cells to flourish and to grow and proliferate, okay? So there are one type. The second type is there are some kinds of infections like TB. TB needs more time and 3 weeks to diagnose TB from the usual or the ordinary cultural conditions. So if I dependent or I, if I have a certain patient with a critical case or critical conditions, and I needed to diagnose if he has TB or not, then I should wait for 3 weeks, and this is way too much time for diagnosis. And maybe in this time be, died and be buried, okay? And I will give the results to his relatives. Is this possible? No. So PCR, will save this time, such a time, and give me the results only in 3 or 4 hours. This is a huge amount or this is, a huge effect on accelerating the diagnosis, the medical diagnosis, and to provide such patients with the proper critical care and so on, right? So this is number one, the use of PCR. Number two is forensic testing. Forensic testing, and this is beyond your curriculum, but you only need to know that PCR is used for forensic testing, okay? So this is about the first part of our section today, PCR. PCR, polymerase chain reaction. It is different from molecular gene cloning, where PCR occurs in vitro, while molecular gene cloning occurs in vivo. PCR has 3 steps. There's 3 steps, to perform denaturation first, then do annealing, then to do extension, okay? And to make use of the synthetic nucleotides to add copies to the gene of interest. PCR, there's another type called RT-PCR, when you are required to duplicate or to amplify RNA sequences. So PCR is used for diagnosis of infectious diseases that are hard to cultivate and they need much time to reach or to give results on their culture like in TB. So this is the first part of that section.